The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and welcome to Lab Manager's Tech Trends webinar series. I'm Tanuja Kopal, contributing editor for Lab Manager, and I will be moderating today's discussion on innovations in CO2 incubators and their uses in cell culture. We are joined today by two technical experts from Panasonic Healthcare and from Thermo Scientific, and they will be talking about some of the new features for CO2 incubators and the factors that you should be considering if you're in the market to either buy or upgrade your incubator. Our discussions in these webinars tend to be very interactive and very informal. So send in your questions and comments, and we will discuss them right after our speakers have gone over their brief presentations. You can type in your questions in the question box that appears on the right-hand side of your screen anytime during the webinar, and I will be receiving those on my end. Today's webinar will also be available for viewing on Lab Manager's website, which is www.labmanager.com. The website also hosts a number of resources such as application notes, white papers, and links to past webinars which you may also find useful. In our closing slide, we will share with you the email addresses of our speakers, so you can reach out to them for a copy of their slides or for further guidance on this topic. You will not be able to download slides off of the web link, the webinar link that we'll send to you. So with that, let me welcome our first speaker today, Deepak Mistry, who is the Senior Manager for Strategic Development and Marketing at Panasonic Healthcare. A molecular biologist by training, Deepak has been with Panasonic now for over 6 years. In his current role, Deepak is responsible for managing the strategic and core marketing efforts for life science, biomedical, and healthcare products. At the same time, he analyzes market opportunities and creates strategic initiatives for new emerging markets. So Deepak was a part of our expert panel on this topic last year, and it's a real pleasure to welcome you back.
Well, thanks so much, Tanya. We're looking forward to it, and I think innovations are really our focus for this presentation. I think we're all looking at different solutions for not only for cell culture, but I think applications that many of our customers experience on a day-to-day level. And just going into it, it kind of got us to thinking where we have a partnership with BD, who, as you know, is one of the leaders in cell culture for consumables and other systems and products. And we thought about on sort of a different solution for customers that, of course, want to grow more, and that kind of gave us thinking to the concept of accelerating your growth. And fundamentally, most customers use a T175 flask, and BD introduced a multi-flask, which is a 3-layer and 5-layer product, which will be able to triple or 5 times your surface area for culturing. So in essence, you're able to increase your surface area for culturing and being able to culture more in less time. And you can see sort of the some of the graphics here that kind of explain really the impact of sort of the traditional method of using a single-layer flask versus a 3-layer flask, and seeing the sort of the cell yield that's demonstrated from there. So kind of really our focus on this type of solution was greater cell expansion in less time. So of course you see optimized cell expansion due to using both products together. In terms of our equipment, you have some major time savings with our H2O2 decontamination technology, which takes 2 hours. And also in terms of consistent environment, we talk a lot about that with incubators. Sanyo has an exclusive dual IR sensor, which helps to recalibrate things very quickly and to reduce sort of that door effect that you have with CO2 recovery. And then as well, for more specialized applications such as low oxygen or physiological oxygen control, I'll talk a little bit about some of the work that we've done in collaboration with Stanford and some researchers on how we're trying to provide some new solutions there as well. And so going to explain some of these concepts, I'm just going to go really quickly and then transition to this video, which is about 60 seconds. So at this moment, I just want to transition and have everyone just take a look at this video, and then I'll continue on with the presentation.
Cell culturing is a time-consuming aspect of your research. Imagine culturing over 3 times more in a single cycle, producing higher yields in less time, allowing you to spend more time on the work that really matters. Accelerate your cell growth with the Sterisonic Incubator from Panasonic and Falcon Multiflasks from BD Biosciences. Used together, these products provide over 3 times the total cell area per incubator, increasing productivity and yield without compromising consistency. And thanks to the Sterisonic Incubator's unique 3-hour H2O2 decontamination cycle, you can decrease downtime between cycles, leading to even greater cell yield in less time. The environment inside the Sterisonic Incubator is more consistent, achieving full CO2 recovery within 2 minutes after openings, and uniform temperatures allow for optimal cell growth throughout the chamber. Visit AccelerateCellGrowth.com today to learn more and to request a free quote.
So I will now go back to the presentation real quickly here. And hopefully you were able to see in those 60 seconds some of the major concepts, which is really greater capacity, higher yield with this type of solution, faster turnaround with H2O2 decontamination at 3 hours, and then also consistent environment with temperature recovery and 2-minute CO2 recovery. And I also wanted to talk a little bit about the multi-flask for people who are unfamiliar with the product. As I mentioned before, a regular T175 flask is just 1 layer. The 3-layer and 5-layer version of the multi-flask allows you to culture more cells faster and easier. So there's less downtime and less protocol work when you're kind of basically doing the work of 3 T175 flasks all at once. The other thing that I think to stress is that in terms of uniform cell growth, even though this is a 3-layer or 5-layer module, the results from BD and from our use in our incubator showed that you still got the same uniform cell growth per layer. And scaling up from single-layer flasks are really, I think, not as advantageous as using a multi-layer system. And then in terms of justification data, you know, what we can see here and what was covered a little bit in the video, you can see the capacity per incubator competitively for the 3-layer and 5-layer flask. Of course, you're going to get 3 times or 5 times the amount of yield when you're using a 3-layer, 5-layer. And the combination of that data allows us to have up to 50% more cell yield when you're using both our incubator and the multi-flask solution together. And the other thing that we worked on very closely to kind of connect closer with end users is understanding that, you know, a cell culture you know, specialist is really working with a variety of different cell types. And a big focus in our solution and really our project here was to kind of show the results for a variety of different cell types. And we created a module here which you call, you know, increase your ROI or your return on incubation. And the focus here really is to show the data for each cell type and showing the cell yield and also showing sort of the benefits that you would have for each one. So hopefully, you know, new customers or that are unfamiliar to this type of solution or want to know a little bit more data that pertains really to the cell type that they're using, we hope this could be a nice tool for them to kind of evaluate on their own. And here is just basically the data for each of the cell types. I won't take too much time on that, but you can see the variety that we have from MSCs to CHO cells to BHP21. And then I wanted to touch a little bit about decontamination. This is a reoccurring theme. We discussed a little bit about it last time. And, you know, Panasonic does have a different technology. We had done some work with a key opinion leader at Stanford, Lee and Len Herzenberg, and they've actually worked a lot on primary lymphocytes, but also as well under physiological oxygen conditions, which I think has been a big trend right now in cell culture, using low oxygen and multi-gas incubators to incubate your cells, and we found that you have a more stable redox, and a variety of other conditions are improved to improve viability going forward in further experiments. But I wanted to highlight some of the benefits of H2O2. One being thermostatic control. Sometimes with heat, there could be some issues with the effect on the rest of the environment. Also, in terms of on a microbiology level, there's a lot of fungal species as well as several other yeast and thermophilic organisms that may be resistant, of course, to heat. And then speaking towards H2O2 and VHP technology, which has become sort of an industry standard in a lot of GMP environments. Companies like Steris, BioQuil have used solutions for entire rooms to be able to decontaminate using hydrogen peroxide. And what's really beneficial is that it is very effective against heat-resistant organisms and really can react to a variety of different species of contamination. But another thing we wanted to revisit was Mycoplasma. We've, looking at a lot of data that's classically out there, a big focus has been that, Mycoplasma is the key contaminant in a lot of labs. And I think a focus for us was to kind of show how our technology not only can decontaminate Mycoplasma, but also completely get rid of it. And it's one of those bugs that are classically hard to sterilize or remove from your environment. And we'll be publishing in June, July, a study that we'll be working with Charles River, who is one of the well-known labs that work with Mycoplasma, and they've identified, it being sort of as well a majority of a contaminant for a lot of cell lines, whether it be in bioprocessing, cell banks, raw material testing, but then also general cell culture. So I think for us, the cross-contamination part of it is, for Mycoplasma, is a big focus for us, and really what we're going to try to do with some of the efficacy testing that we're going to do is to show not only how well it works, but also the speed at which it works, and then also being able to talk a little bit about why decontaminating an incubator, with it being such a small area, when you have a bug like Mycoplasma, why it's so important to not only decontaminate it in quicker fashion, but also being able to eliminate the bug quicker from the environment. And I think this is where there's a point where I revisited from the last webinar is regardless of which decontamination technology that you choose or prefer, I think what really should be focused on when you're looking at the technical papers or the white papers for these type of technologies for efficacy, make sure to look at the organisms that have been tested. There's a lot of key contaminants like Mycoplasma, Staph aureus, various yeast and mold species. Stereothermophilus is a big one because it's thermophilic. So our really advice from a technical level is, regardless of which technology you choose, make sure to really look that the efficacy is really proven for some key organisms that you would actually experience in contamination. And last, I wanted to touch on some of the work that we'll be publishing at the International Society for Stem Cell Research. That will be June in Yokohama. And we've been working, as I mentioned, with a key opinion leader, Lee and Len Herzenberg. And they've been very notable. They've won sort of the Kyoto Prize for their work on physiological oxygen levels using primary lymphocytes. And they've come to that point where working at physiologic oxygen levels, they've been able to get better viability in their cells, have better redox states, and really in turn where that really comes into play is when you're going further down the line in your research protocols, whether it be maybe in cell therapy or other types of studies where you need to have those cells more viable. And in the past, I would say 3 to 5 years, there's been sort of a plethora of research that has mimicked sort of the same concept here of culturing a variety of cells from HIV to mesenchymal stem cells, human stem cells. And so it's really been a big trend. A lot of researchers have really kind of embraced this idea. And so in summary, we've been actually working on a solution for these customers that are working at physiological oxygen levels. What most people understand is when you enter an oxygen level that you want to prefer on your incubator, whichever incubator you have, you hope that it's actually providing that oxygen level and CO2 level to your cells. But what we've seen in a lot of studies with BD and with us combined and with Stanford is that what's really going on in that cell culture plate could be a little bit different, and there could be some variation. So we were working on a solution that not only will let us monitor the oxygen levels that are actually going on within the cell culture plates using a oxygen reader dish, but also what we wanted to do is incorporate a on-site nitrogen generator. Most labs may or may not have the ability to replace tanks as readily, and if they have something on site, it may not be as consistent as having an on-site generator. And there's could be some big advantages, maybe even in terms of savings, cost-wise, to going with this model. But we will be presenting sort of the data on this sort of real-time physiological oxygen control. I will have to say one of the funniest things I've ever heard in the lab is that a lot of the researchers on that floor have now called this system the perpetual motion machine for oxygen delivery. So we're excited to present the results in June at the IACCR, and I think in summary, we're happy to help with new innovative solutions for our new cell culture customers, and I'll look forward to the discussion later on.
Great, thank you, Deepak. If you have any questions for Deepak, feel free to type it in and send it over. I would also like to remind you that today's webinar will be available for viewing on Lab Manager's website for at least 6 months. And the link to the archive webinar will be emailed to all of you sometime next week. We will also share with you the email addresses of our speakers after our next presentation. So you can feel free to touch base with them directly if you're looking for a copy of their slides. So our second and final speaker today is Mary Kay Bates, who is a global cell culture specialist with Thermo Fisher Scientific, where she provides cell culture expertise to both colleagues and customers. Before joining Thermo Fisher, she managed technical support and distributor relations for a small company producing cell culture reagents. Mary Kay has spent many years working in the academic and an industrial cell biology lab, focusing on gene therapy and cancer research. And we are glad to have you back with us again, Mary Kay, for the second round on incubators discussion. Welcome.
Thanks, Tanuja. I appreciate the introduction, and also thanks to Lab Manager for providing the forum today and for inviting Thermo Scientific to participate. As Tanuja indicated, I'm relatively new to Thermo Scientific, and I joined the company after nearly 20 years in cell and molecular biology research in both academic and small biotechnology labs. So I'm part of the scientific team at Thermo. And if you are like I was, you probably just want an incubator that you don't have to think about. But the thing is that understanding of cell biology has come a long way in the last 10 years. And so this is not really your parents' cell culture anymore. So the main reasons to upgrade your CO2 incubator are focused on your cells. So as an introduction, I just have a few words about my company, Thermo Scientific. We manufacture 2 flagship incubator brands, Forma and Harricell, and both brands have decades of quality and innovation for CO2 incubators. And this long experience has given our designers and engineers many collaborations with people all around the world doing cell culture. At Thermo, we believe that your next CO2 incubator should have not only the very best quality, but a selection of choices that allow you to match your incubator with your needs. Those include the choice of traditional water jacket heat or direct heat, which allows you to have automated sterilization of the chamber. And that's just one of the contamination control options we offer. Another option I'm going to talk about is variable oxygen control for physiological culturing. We've been making incubators with oxygen control since 1979, but as I'm going to show you, newer research means this is a great reason to upgrade now. And we offer your choice of thermal conductivity or infrared CO2 sensors and a wide range of incubator sizes to best fit your lab and your applications. And we offer monitoring technologies to tie with your building alarm systems. And finally, all of these choices should make it easy for you to put your cells in the incubator and just leave your worries at the door. So why should you upgrade your CO2 incubator? Well, there are a lot of choices now that I didn't have when I was in the lab. But more importantly, cell culture is changing. We all used to work with long-established cell lines where some, like HeLa cells, have been manipulated so much they're almost their own life form now, and they really aren't good models anymore for in vivo conditions. Research and, to some extent, development is now often performed in more physiological cells, those that are more recently removed from the body or tissue. And these cells don't have the hardiness of traditional immortal cell lines. So I'm talking about cells that are more difficult to culture and grow: primary cells, neural cells, immune cells. And with stem cells, we are often trying to keep them from differentiating, trying to keep them pluripotent. So one new development that I think is really exciting is growing cells in 3D, and how that facilitates more physiological communication among the cells. And so along with growing cells that are more fragile and harder to keep growing, there's also more awareness of the perils and risks to biological and chemical contamination. So that gives us a greater understanding of tissue microenvironments and how conditions vary depending on where the cells are located within an organ or tumor due to new research. So I mentioned risks of contamination. This has always been a problem in cell culture. Every person working in a cell culture lab, experiences contamination occasionally because microorganisms are our constant companions. As just one statistic, a recent study found that we carry, on average, 10,000 microorganisms per square centimeter on our skin, which is kind of scary. And in line with that, a study from 2002 found 23% of research lab cultures carried Mycoplasma, but that's not limited to academia, as a study from about a year and a half ago found 8.5% of biotechnology and pharmaceutical cultures were contaminated. And since these were from companies that routinely test, the real numbers are likely even higher. So antibiotics, when you routinely use them, actually mask the problem by hiding low-level infections, and they do not work against Mycoplasma. So it turns out that labs that routinely test for Mycoplasma contamination have the lowest incidence because the problem can be isolated and eliminated before it spreads. But another easy way to reduce your chances of contamination is to use an incubator with a design that itself helps prevent contamination. Now, there are quite a few ways that your incubator can control contamination. Some direct heat incubators now come with an automated high-temperature sterilization cycle that eliminates any life from the incubator interior. As with all sterilization, it's dependent upon not only the temperature, but also the amount of exposure time. This means that you no longer have to spend time and money cleaning with chemicals or disassemble the incubator parts, then put everything back in the autoclave, and then carefully put it all back in the incubator, and you're trying all the while not to reintroduce contamination. Now, different manufacturers offer different methods, so as Deepak said, the only way to really compare them is to check independent results showing elimination of text organisms, including resistant spores, to make sure that the time and temperature combination is effective. Now, there are also a couple of choices of easy ways to prevent contamination in your incubator before it even gets established. So you can start with a clean incubator and keep it clean. And a very popular option is a HEPA filter, which will continuously filter the chamber air, removing unwanted contaminants. But here again, different manufacturers feature a HEPA filter, and the differences lie in how the HEPA filter is designed into the incubator. So check the air exchange rate. How often is all the chamber air cycled past that HEPA filter? And is the HEPA filter just on the gas line, or is it actually inside the incubator chamber? So the other cool choice for contamination prevention is solid copper, which is itself antimicrobial. A copper interior for your incubator requires no maintenance at all, and it means you don't have to use smelly germicides to clean the incubator. And these germicides can leave residues and fumes, which themselves can be hazardous for your cells. And copper will last the life of your incubator. You never have to replace it. Now I want to show you some data showing how well copper works. It turns out that the less copper content you have, the less effective it is for killing microorganisms. In fact, the U.S. Environmental Protection Agency tested all kinds of copper, and they certified only alloys with more than 60% copper content as being antimicrobial. Now this graph shows over 6 hours that methicillin-resistant Staphylococcus aureus bacteria, or MRSA for short, thrive on stainless steel. And you can see that in the blue. So there's no reduction of them over 6 hours. But if you look in the red, solid copper will kill 10 million of these supergerms within 90 minutes. And as you reduce the amount of copper in the alloy, so the green has 80% copper and the brown is 55% copper, the killing of the MRSA germs is delayed. So less copper content means less antimicrobial effectiveness. And it turns out that tarnished copper is more effective than bright, shiny copper, and only alloys that have more than 55% copper will show tarnish. This graph shows time over 60 minutes, and it shows that 99% copper that has tarnished, that's shown on the blue line, kills off more than 10 million E. coli much more effectively than the same copper where the tarnish is removed, and that's shown in the red line, which is higher up on the graph, showing no reduction of the E. coli, or very little. And so lower copper content plus no tarnish really decreases the antimicrobial effect. And the copper actually gets better with time. Real copper will tarnish, and the oxidation of the tarnish contributes to the lethality of the copper. So if it doesn't tarnish, it's basically ineffective at killing microorganisms, as the U.S. EPA has determined. Now, the copper ions do not become airborne, just like you cannot easily wipe off the tarnish. So the copper is safe for your cells. It only kills on direct contact with microorganisms. And there have been exhaustive tests on dozens of microorganisms that are killed by copper, including bacteria, fungi, spores, viruses, even prions are affected. Now, I didn't have copper in my lab, but I've talked with quite a few people who do have copper incubators, and they love them. For example, one principal investigator told me how since they got the copper, they never have any contamination problems at all, and the lab next door still struggles with it in their stainless steel incubators. He said his lab hardly ever cleans the incubators anymore, so the copper saves them a lot of time and money that they used to spend on cleaning the incubator and fighting contamination. And now I'm going to spend just a few minutes talking about an aspect of cell culture that is really fascinating and exciting to me, and Deepak touched on this as well, and that relates to the incubator in terms of variable oxygen control. Now, most cellular functions are regulated according to oxygen concentration, mostly by hypoxia-inducible factor 1. This means that the amount of oxygen that cells experience can affect everything in the cell: metabolism, proliferation, morphology, disease progression, and so on. Most cells in vivo experience an oxygen gradient depending on how far they are from the bloodstream, and most cells normally experience hypoxia or low oxygen far below atmospheric oxygen, which is 21%. So I have some examples here, including lungs with 14% oxygen content. Liver, heart, and kidneys are more hypoxic with concentrations varying from 4 to 12%, and so on. So my message here is that to best simulate conditions in the body, you might consider choosing an incubator that offers a tri-gas option, which reduces oxygen concentration by supplying nitrogen gas. Now, let me show you an example of how oxygen affects the general growth of primary cells. This example is in newly isolated primary mouse embryonic fibroblast cells. If these cells are cultured in normal atmospheric oxygen of about 20%, you can see on the graph, over time, the cells stop growing and dividing about 28 days after they were isolated from the mouse. At that point, the cells have a distinctive appearance and gene expression where they are no longer actively growing. They shut down and enter senescence. At the same time, the cells show oxidative stress with DNA damage similar to what we see when we treat them with hydrogen peroxide. In contrast, when the cells are cultured in 3% oxygen, as you can see in the new line I put up on the graph, the cells grow faster. They are dividing in only 25 hours compared to dividing in 38 hours at 20% oxygen. And the cells kept growing and dividing out to 60 days, much longer than the 28 days we see at 20% oxygen. These cells at 3% showed a normal appearance. They had normal gene expression, so no senescence. and they had no oxidative DNA damage. So with just growing the cells at physiological oxygen, the cells are much, much healthier, they're not under stress, and they grow like they normally would in vivo. So I have just one more example of hypoxia and how it affects cells. Hypoxia is also important in cancer, and this is becoming increasingly recognized. Research shows that tumor microenvironments have an oxygen gradient. At the outside of the tumors, next to the capillary blood vessel, which is bringing oxygen, the tumor cells are expanding. And as you go toward the center of the tumor, the oxygen concentration decreases, and you find hypoxic cells until you get to the very center, and the center cells with little to no oxygen are necrotic. So these cells in the center are very different from the expanding tumor cells on the outside of the tumor, where there's a lot more oxygen. And these necrotic cells are linked to resistance to chemotherapy drugs and radiation, and they are linked to continued tumor progression. So this is an example of how the cells are in different microenvironments with different oxygen concentrations depending on where they are located in the tumor or organ. So the take-home message here is that cells that are cultivated at low oxygen better mimic in vivo conditions. So just a few conclusions. Now is a really great time to consider upgrading your cell culture incubator. There are more choices than ever that give you more control over your work. And these choices include easy-to-use, hands-free contamination control, including high-temperature decontamination, HEPA filtration, and solid copper interior and parts. Remember to compare effectiveness data from different manufacturers. And consider an incubator that offers variable oxygen control. Cells cultured at hypoxia exhibit faster growth, lower stress responses, and less oxidative stress. Solid tumors and organs have different microenvironments with varied oxygen conditions depending on the distance that the cells are from the arterial blood vessels. So cells cultured under hypoxic conditions are more predictive of the in vivo state, so they're better models for functional biology, disease progression, and more. To learn more about CO2 incubators from Thermo Scientific, visit our website at thermoscientific.com/CO2 incubators. There you will find lots of information and resources, including application notes, magazine articles with more information about contamination and oxygen, and notes explaining our engineering designs and how they work. I want to thank you all for your attention today, and the next slide will show Deepak's contact information and mine. Feel free to contact me directly with cell culturing questions. Thank you.
Great. Thank you, Mary Kay. Yep, and the email addresses are right there on your screen. If we have a few questions that have come in, but if you think of other questions after the webinar, feel free to reach out to our speakers directly. So, a couple of questions about the copper versus stainless steel, I guess. So, Mary Kay, maybe you should just sort of help define or compare and contrast the two. What are some of, I know you went into some of the details with the advantages, but do the copper incubators cost more while you have to spend less time cleaning it? You also mentioned something about direct contact with the organisms, only then it will clean it. So does that mean a copper incubator has all its parts made of copper, including the shelving and anything else that comes in contact with the cell culture plates? Do you mind going into some of those details for us?
I appreciate the opportunity, Tanisha. Okay, so let's just first talk about maintenance. Copper doesn't require any additional maintenance than stainless steel. You can use the exact same things for cleaning the incubator. The difference is you don't have to clean the incubator. I mean, obviously if you're spilling some cell culture media on a shelf, you want to wipe it up just like you would in stainless steel. But you don't have to take out all the parts and stick them in the autoclave because the copper itself is antimicrobial, and that includes fungi as well as bacteria and spores and so on. We do often get questions about, is the copper going to affect my cultured cells? And it's a solid surface, so the copper doesn't become airborne. Just like, I don't know if you've ever polished silver, for example. The silver tarnish in general doesn't wipe off when you rub it with your fingers. You need to use a tarnish remover. So just like that tarnish doesn't come off easily, it's part of the copper intrinsically that those copper ions also do not become airborne. So as long as your cells are in the culture dish and they're separated from the actual copper by the plastic in the dish or the flask, they are not affected by the copper. It's only the microorganisms that make direct contact with the copper by landing on the copper that kills off the microorganisms. And it's interesting, we don't exactly know the mechanism yet for how the copper kills. We know it kills on contact and that there's membrane blebbing and that the copper ions then, once the cells are compromised, the microbial cells are compromised, the copper immediately starts attacking the nucleic acids, especially DNA. But we don't know exactly what's happening on the membrane level. Is it taken up actively into the cells, or is the copper somehow just causing the membrane to disintegrate and bleb? Let's see. I think another question that you mentioned, Tanuja, was is the entire interior copper?
Right. Mm-hmm.
On the hair cell incubator, yes, the entire chamber is copper. The shelves, the fan even is copper, so you don't have to worry about cleaning the fan. And then for other incubators, there are parts available, so you can get copper shelving and copper water pan, for example. And that's if you think about the fact that most bacteria are, even though they circulate a little bit, they also fall from above, right? That's why you have lids on your culture dishes. They're not airtight, and if you, for example, have a fly in your incubator, theoretically it could crawl up inside the lid, but that doesn't work that way with microorganisms. They fall from above. So as long as your shelves and your water pan are not harboring microorganisms, it's a little less important that the walls, for example, are copper. So if you wanted to, you could add in copper parts later. But for the very best effectiveness, for the very best protection, you'd want the entire interior parts to be copper.
Okay.
Did I miss anything, Tanuja?
Does it cost more?
It does cost a little more, but when you think about the peace of mind that you have, the people that I talk with who have copper, they just kind of go on and on about, you can hear it in their voice, how happy they are that they don't have to really clean the incubator anymore. The Whitehead Institute, for example, they work with stem cells pretty much exclusively in some of their labs, and all of their incubators are copper because those cells are so susceptible to infection. And the same with primary cells, that the copper just gives them the peace of mind that they don't have to worry about the contamination. And the labs next door, like that guy that I talked about earlier, his lab next door has stainless steel, and they're just always fighting contamination. But his lab with the copper, they don't worry about it.
Okay. Well, appreciate all that feedback. Deepak, did you have anything to add to this copper discussion?
Yeah, I think that we do get that question that comes up a lot, and I think Mary had some good points there. I think one thing that I think researchers should always be mindful of is not only whether the incubator has copper, but also to kind of speak to the manufacturer about what type of testing that has been done for their particular copper and whether or not it's been effective against species other than bacteria. Because as we've seen, we have a lot of issues with Mycoplasma. There's a lot of contaminants predominantly for fungus, yeast, and mold. So I would encourage that when you're evaluating any type of chamber with copper to look at that, because I think some of the information from the EPA focuses on E. coli only. So I think but it also depends on looking at the study that the manufacturer has on their particular type of copper. And I would say the second to that is that the chamber is something, of course, that you need to be sure of to decontaminate. But a lot of times, I think as Mary mentioned, the contamination comes from the air. When you open the door, you may have a hood or a vent, and bacteria can come in through the air and take into the chamber and may not have on contact right away to the chamber surfaces. So, I would say look to see what technologies people have to also prevent that. Our incubators have an active background contamination control where when you open the door, the UV light shines in the water pan, which is shielded, and it scrubs the air continuously. And there's some differences in terms of how that kind of technology works compared to some other background contamination control. So I think it's really good to kind of take an in-depth look at what your environment is and really what technology or combination of technologies are best suited to combat any type of proactive contamination event.
No, absolutely. And I think this webinar is the first step towards that, comparing and contrasting different technologies and trying to figure out the one that works best for you. So the next question is about the sensors. Which one is better? Would you recommend a TC versus a IR? Any thoughts on that?
Would you like to go first on that? Tanisha, would you like me to go first on that?
Doesn't matter.
Okay. Is that okay with you, Deepak?
Yeah, no problem.
Okay. One of the things that Thermo offers that some other manufacturers no longer offer is the thermal conductivity sensor. And the reason we still offer it is because it's still super popular with people. It's reliable. The reason that it's reliable is that it has been around for decades, and the technology is well established. It's a very robust sensor. You'll often hear nowadays that the infrared sensor is much better, that it responds faster, it's not subject to humidity, and it is true that it doesn't. The TC sensor doesn't recover quite as quickly, but the thing is, it's going to last the life of your incubator. The infrared sensor it's basically a light bulb, and like any light bulb, it's going to burn out over time, and then you have to replace it. So people who don't want to spend that extra, that extra money, if they don't need the quite as stringent of a response, they're happy with their TC sensor. Like I said, it's still a hugely popular sensor for Thermo Scientific, and that's why we offer both choices, because people want both.
Okay. Deepak, your thoughts?
Yeah, I mean, I think this is another, feature in an incubator that you have to look very closely on. I think TC was an industry standard for a long time, and it had, I think, reliability is really based on which manufacturer you choose, and you really need to kind of, do some research on that. And I think one good step is to see the recovery rates with each type of sensor. And it has been shown, though, that IR has better performance in a lot of cases with a lot of different manufacturers for CO2 recovery. But I would really focus on trying to find out really what the reliability is, because a TC sensor could sometimes be less expensive and was something that you were used to. But on the long haul, you may want to consider a technology like IR. We offer a dual IR, which is sort of, the next level of this, where you're going to have much more precise CO2 control, and it also allows you to recalibrate on the fly. So when you're having a lot of door openings, you're going to have a lot of issues recovering. And having this feature, you're able to auto sample, and the actual component itself has no moving parts. So essentially, in one sense, it would be more reliable. So what we've seen from our customers is they have trended towards IR more readily for the precise control. But I think what it comes down to is looking at how the actual sensor is made. Our sensor is made with a ceramic heater with a light source, and that can obviously transfer to more reliability in the operation of the sensor. So my advice would be to, as researchers, when I was in the lab, I wasn't an expert, of course, in all these type of technologies, but I think it's come to that point where if you're putting an investment in an incubator or several incubators, it's worth getting a good explanation from the manufacturer on how those sensors are developed, why they're more reliable than the other, and also why they're more precise. And before looking at really the cost differences, because at the end of the day, you don't want to buy a technology or an incubator that you may need to either replace, or you have to replace the sensor, because some sensors for some manufacturers could be expensive, and it just becomes an issue. So I would say research, 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 you know, when evaluating these technologies.
Okay. Deepak, I think this goes back to the comment that you just made regarding the UV light. So the question is, for the UV, wouldn't it be you know, dependent on the time of exposure? So in this case, how long does the water in the pan needs to be exposed to make sure that you're you know, effective in killing all the microorganisms?
It's a very good question. You know, in our white paper, we've shown there's a variety of time of exposure for different types of organisms. I think what's good, though, is that the UV function within our incubator is a sort of a proactive control. So it's not the main decontamination method. You know, that's why we have H2O2 that can be done in 3 hours. So as far as, you know, exposure time, I mean, like I said, there's a variety of different time levels for each one, and then, you know, I think usually on general for most of the classic organisms, the 5-minute exposure is enough for at least the airborne. Outside of that, we would recommend running the H2O2 cycle. If you have an incubator that, you know, doesn't have that feature and only has UV, then we would recommend just doing a 24-hour UV cycle, which really could address, I think, every organism that you would encounter. And I would say for the major organisms that you have contaminants, you could do it at 12 hours.
Okay, sounds good.
I just have something to add on that, Tanuja,
sure.
if I could.
Absolutely.
Thank you. Thermo Scientific actually put UV lights in our incubators a long time ago, and we stopped offering that because the customers felt like it wasn't really doing anything. And it's kind of something that I think at first blush we all think UV is great because everybody uses it in their biosafety cabinet. And it's just a different situation inside the CO2 incubator, especially if you're trying to decontaminate either the water or the air. And that's because in the biosafety cabinet, you're decontaminating an immovable surface. It's just sitting there and the light is shining on there. What we find in the CO2 incubator, and this has actually been published, that UV light is pretty much ineffective at high humidity when the humidity is greater than about 70%. So I think it's great that Sanyo has revived this technology, but we kind of feel like it's not real effective in the CO2 incubator.
Okay. Well, thanks for your candid feedback. There are a lot of questions regarding the high temperature sterilization slide that you put up. So let me go through them, you know, one by one. With regards to that slide, you said that there was no more disassembly of parts needed. So I think people need some kind of clarification in the sense that what if you have an IR sensor? You know, are you saying that things can stay in the chamber during that sterilization cycle? You know, what about the HEPA filters? So can you, I guess you probably need to go into some details regarding that high temperature sterilization mode.
Sure. Yep, and it depends on the manufacturer and it depends on the sensors. At Thermo, our sensors are manufactured exclusively for us, and so they are very robust. So depending on the temperature, for example, the Haircell incubator has a 90-degree moist heat decontamination cycle. And the reason that that's a moist heat is that we can use a lower temperature, and our sensors, both TC and IR, will withstand that temperature. And because it's moist heat, it's more lethal and more penetrating than dry heat. So if you're going to use a dry heat, then you would need to use a higher temperature. So, for example, the Forma incubators use a 140-degree cycle. And both of those have been proven with multiple test organisms to kill off, you know, all of those microorganisms. And then the other thing I would say is that it's important when you're evaluating those high heat cycles to make sure that the incubator actually reaches that advertised temperature, because it is the lethality is dependent not only on the temperature but also the time of exposure. So it's a combination. If you're If you're doing a lower temperature, you might need a longer time. If you're using a higher temperature, then yeah. You asked about HEPA filters, and right now the technology is such that the HEPA filter will not withstand the high temperature, but we're working on that, and that should be coming in the coming years.
Okay, and then with this heat sterilization, you don't have to wipe everything down afterwards? Is that how it works?
Nope. If you think about it's not the CO2 incubator is not an autoclave. You don't have the pressure, so that's why it takes longer. You're just doing it with heat. So, but the similarity is that it's essentially sterilizing the interior. So you wouldn't have to wipe anything down. There's no, with the high heat, there's no residual chemicals that you need to use. One of the things that you see published a lot now is how cells are susceptible to volatile organic chemicals from bleach, for example, from acetic acids and peroxides, and so all of those fumes can stay in the incubator and then really affect the growth of your cells. That's one of the reasons why, in terms of HEPA filters, we also offer ones that will collect volatile organic chemicals in the HEPA filter and protect your cells in that way. And those are actually becoming very popular with people using stem cells and incubators for in vitro fertilization, things like that.
Okay. Well, actually, Deepak, that brings me to the next question, which is on the use of hydrogen peroxide for decontamination. So what is the concentration of H2O2 that's used, and how does this affect the cells, if it does?
Well, I mean, the decontamination method uses 6% H2O2. It doesn't affect the cells because the process happens after, when you're not culturing, and the UV stage resolves it with absolutely, a residue that's basically inactive. And going back to, I think, the last question, I think determining whether or not which method you use, I think if you're concerned about time, I think you have some advantage with H2O2 being able to do that in 3 hours. And I think second, really evaluating what types of organisms, again, that you're encountering as a contaminant. And if you're going to have a situation where you're going to have some type of thermophilic organisms, or or some contaminants such as yeast and mold, H2O2 has been shown to be very effective against things like that. So I would say that's just something that you need to keep in mind.
Okay, and you mentioned it takes about 3 hours on an average for the H2O2 decontamination?
Correct, yes.
Okay, so then what about the high temperature decontamination? How long does that take? And do the cells have to be removed when that decontamination is ongoing?
That's a good question. I actually asked that when I first learned about this too, because I didn't have high heat decon in my lab either. I had to clean it just like everybody else. So I have mentioned that the effectiveness of the decontamination is a function of time and temperature. So sometimes if you have a higher temperature, it takes less time. The hair cell incubator, which has the lower temperature, it does take a little bit longer. I don't recall the exact time overall, but I mean, pretty much any time you're doing these processes, what you're going to be doing is taking your cells out of the incubator because they won't withstand the heat, and putting them in another incubator and running it overnight. For most people, that works out great because it's just the end of the day, and then you come back in the morning to a nice, clean, sterile incubator. So it's because it's not an autoclave, it does depend on time.
I see. Okay. And then with regards to like the inside of the incubator, do we have incubators now where different zones can have different temperature and humidity controls?
That's actually something that's very difficult to do because when you have different zones, each individual zone will require its own sensors. So each individual section will require its own CO2 sensor, humidity control, because what you don't want to see happen is when you open the door of one chamber that all of the chambers get that surge of CO2 and heat. So in other words, if you have cells in one section and then you open the cells, the other section to access those cells, then the cells in the first section might be affected if you don't have separate sensors. So while there are some incubators now, especially for in vitro fertilization processes that offer different chambers, most of them have only the one sensor for the whole unit. So it's a concern that your cells are going to get an additional boost even if the door to that section hasn't been opened. That's something we're working on at Thermo.
Okay. Deepak, your thoughts?
Yeah, I mean, I agree with Mary. It's somewhat of a, I think there's some incubator hotels, but those are pretty expensive that I think are for IVF. One solution we have had, like for customers that need this, and we've seen this in IVF, is we have a triple stack incubator, which is only each chamber is 1.7 cubic foot. So, essentially you're able to, when you're working with maybe, single patient samples or just something individually, that's one solution that I've seen some customers gravitate to, for having like, 3 different zones, and being able to sort of have 3 chambers cost effectively because they're all, smaller size, and also they have, better control under that smaller environment.
Yeah, and that's a good point, Deepak, because there are very small incubators that are just tabletop things that are offered. One of the things that Thermo offers a lot of is different size choices, and so I do like, we have a 1.4 cubic foot incubator called the MIDI, and I like that, especially for use as a quarantine incubator. If you're worried about you're borrowing cells from a colleague and you want to quarantine them to make sure they're not contaminated with mycoplasma that can run through all of your cultures. So a quarantine incubator is a great idea for that reason. Good point, Deepak.
Yeah. All right. I mean, speaking of mycoplasma, have the copper incubators actually been tested? I mean, do they just decontaminate or do they prevent mycoplasma from growing?
The thing about mycoplasma is it is a human pathogen. So, I talked about how we all carry, on average, 10,000 microorganisms per square centimeter on our skin. And so it is a human pathogen. It can come in with humans. And so that is one of the things that we looked at, particularly with the copper. And mycoplasma are indeed susceptible to the contact killing by the copper. And there are lots of publications. While I only showed the MRSA and the E. coli on the experiments that I showed, there are hundreds of publications on copper, and there's actually even a website for them called antimicrobialcopper.org. And one of the reasons there's a website for this is that copper is becoming so trusted that many hospitals are starting to put it in on touch surfaces, so doorknobs and handrails and things like that, to help reduce hospital-acquired infections of things like mycoplasma, which Mycoplasma pneumoniae is one of the species of mycoplasma, and things like Pseudomonas aeruginosa that are nosocomial infections. So the copper does work on mycoplasma. It works on fungi. It works on viruses and lots of bacteria. I haven't yet found really any species that are not susceptible to copper.
Okay, what about mold?
Mold, indeed, will be killed by copper.
Okay.
And if you think back, many of us used to stick pennies in our water pan to keep fungi and mold down. It doesn't work so well anymore because copper pennies tend to be almost no copper anymore. I've heard of people putting speaker wire in their water bath. So.
Okay.
Tanya, to add to that, I mean, I think, once again, I think copper is it's been there for a while, and what I would say is, and I agree with Mary, there's hundreds of articles on the effectiveness of copper, but I think what is important should be important to a researcher is to find out specifically from the manufacturer on testing that they've done for their exact copper that they have in their incubator, and show the effectiveness of it against mycoplasma and other types of species. From our standpoint, UV does work on mycoplasma. We have enough data on that, and as well as H2O2. But I think one other thing to think about is, things like mycoplasma, as Mary said, could be, coming from your hand, or it could be airborne, and so sometimes you may not take contact onto the actual chamber or the walls. It might be in the air within the chamber, And I think that's where you have to kind of actively look at, with HEPA filters, there's a certain, I think it's about 0.3 micron. And, Mycoplasma and things like that are smaller. And so that could be sometimes something to consider, and be able to, I think the other issue with Mycoplasma is that it's difficult to detect. So I think, I think those are things that need to be, I think researchers should be concerned about. If they know that this is a known contaminant, they need to know what technologies, have proven to work specifically for that manufacturer, and then also look at being able to look at, how they can detect it, because a lot of times you can go to attacking a problem, but with not knowing what it is or what species it is, it can be really difficult in handling the problem.
Yeah, and I like what you said about manufacturers doing their own testing. We obviously do a lot of testing at Thermo, but what we have found is that people prefer to see independent outside experiments. So while we can and have done a lot of testing of our own, we tend to prefer outside independent testing that's non-biased. So we're using solid copper, and you can't get any more antimicrobial than that, and that's proven by outside testing, not our own testing.
Okay. Well,
yeah, agreed. I mean, I think the testing that I was talking about as well is about independent testing.
All right.
Thank
okay.
You.
Well, yep. So this brings us to the end of our questions, but I'm sure there will be other issues and other questions that crop up. So you have both our speaker email addresses on your screen. I'm sure they both will be happy to help all of you out with your specific concerns and questions. So on behalf of Lab Manager, I would like to thank Panasonic Healthcare and Thermo Fisher Scientific for their participation and support of this event. My sincere thanks again to both our speakers for putting in the time and effort to make their presentations very relevant, very scientific and informative. The webinar will be archived on our website for at least 6 months. So do visit us online to review this webinar, to check out past webinars, or to get more information about upcoming webinar topics. Thank you all once again for your time and attention. Until next time, goodbye and good luck.